Hi everyone, this is Dr. Omyu from University Sultan ZAINAL ABIDIN, and my title for today is APIGENIN, LUTEOLIN and QUERCETIN modulating natural killer cell cytokine production. According to the latest data, lung cancer is the most common nowadays, and in Malaysia it is the third most common cancer. Although various treatment options for lung cancer are available, and yet the number of the case as well as the mortality rate due to lung cancer is still very high. And alternative therapies such as immune cell mediated therapy are nowadays considered as an alternative options to treat lung cancer. Natural killer cell, one of the immune cells, are the third highest lymphocyte in the circulation, and they use multiple mechanisms to kill infection and cancer. One of the most popular mechanisms they use is intracellular cytotoxic granule secretion as well as cytokine production. The immunomodulation by plant-derived products is one of the areas of interest currently for cancer immunotherapy, and understanding the subcellular mechanism of how NK cell mediate the cytotoxicity will be beneficial to develop novel NK cell based chemotherapy. There are thousands of flavonoids available in nature, and these flavonoids are subdivided into 7 classes. The apigenin, luteolin belong to flavone and quercetin in flavonoid group. These 3 flavonoids are selected for our study. The plant-derived flavonoids show various mechanisms in helping immune cell. One of these is immunomodulatory reactions, and many researchers has documented about relationship between the flavonoids and immune cell. Based on our previous study, the NK cell killing to the lung cancer was significantly achieved by activation with different concentrations of apigenin, luteolin, and quercetin, and we also found that the mechanism for this scaling is linked to perforin and granulocyte secretion from the NK cell. Although the NK cell and cytokine secretion are linked to various immunomodulatory reactions, and yet the molecular mechanism of how the flavonoids affect the immunomodulatory action on NK cell still has to be elucidated, and thus we aim to demonstrate the effect of flavonoids compound on NK cell cytokine secretion. In this study, we use NK92 cell line and the commercially available apigenin, luteolin, and quercetin. And the 4 cytokines we did measure are interferon gamma, interleukin 2, interleukin 4, and interleukin 10. Their level are estimated after NK cells were treated with different doses of 3 flavonoids and activated with lipopolysaccharide. For statistical analysis, we use one-way analysis of variance ANOVA method followed by Dunnett's multiple comparison test using the Graphite Prism 6.0 program. The ELISA result showed that only luteolin 25 microgram per ml concentration significantly increased NK cell interferon gamma secretion. However, in case of interleukin-2 secretion, except apigenin, luteolin and quercetin in all different concentrations produce the significant elevation of interleukin-2 from the NK cell. The 3 flavonoids, apigenin, luteolin, and quercetin, show no significant changes in interleukin-4 and interleukin-10 and secretions from NK cell after treatment. Cytokine production is one of the key features for the immune cell to destroy the attacker cell, and in our study we have found that quercetin and luteolin treated NK cell significantly increased the type 1 cytokine production, especially luteolin 25 microgram per ml treated NK cell significantly produced both interferon gamma and interleukin 2 secretion in our study. Other study has shown that lymphocyte interferon gamma secretion is interconnected with interleukin 2 level. In our study, With the treatment with luteolin 25 microgram per ml concentration, the NK cell produce four times higher level of interleukin-2 level compared to the normal untreated control group. So the amount of interleukin-2 produced by this luteolin concentration is sufficient enough to bind to the interleukin-2 receptor and then subsequent interferon gamma secretion from the NK cell. However, the quercetin-treated NK cell also produce a 2 times higher level of interleukin-2 secretion. It is assumed that the amount produced by quercetin-treated NK cell is not sufficient enough to stimulate interferon gamma secretion. Surprisingly, APIGENIN showed no significant changes in interleukins level in our study. So it is assumed that the mechanism APIGENIN use may be different from other two flavonoids. Other studies have shown that APIGENIN use different molecular mechanisms such as NK cell receptor activation and intracellular kinase stimulation. And another study also documented that there are different cytokines they use other than the four cytokines that we did measure in our study. And another possible mechanism for the APIGENIN insignificant elevation of interleukins could be the presence of hydroxy group in their ring structure. APIGENIN, which has three OH group as you seen here, and compared to luteolin 4 hydroxy group and quercetin 5 OH groups in their ring structure. The presence of OH group is crucial in binding to their receptor or activating the intracellular mechanism. So we assume that it can be due to the presence of different OH group which might differ the action of luteolin and quercetin than the APIGENIN. We concluded that luteolin-treated NK cells show the promising result, and luteolin should be considered as an adjunct treatment to current conventional method in immune-mediated disorders, including cancer. Further studies should focus on the effects of flavonoids on NK cell receptor expression or intracellular kinase production. These are the references for our study.